This is a single-cell organism called Saccharomyces cerevisiae. That name is far too long, so we're going to call him George. George is a type of yeast that's been used since the ancient times in the making of wine, bread, and beer. However, we'll find that he gets quite a bit more interesting if we quickly open him up. Ow! Sorry, George. Inside George, there's a little ring of DNA called a plasmid. The DNA in this plasmid contains the code for a whole bunch of proteins that George uses in his day-to-day life. Now, little does George know, a scary, ruthless, conniving creature many thousands of times larger than him has hijacked his plasmid in order to use his body to make its own resources. The horrible beast that could do such a terrible thing as this to our friend George? A human. That's right. Since 1973, humans have been able to effectively hijack the plasmids of bacteria like George and use them to produce proteins and hormones that previously would have only been found inside other organisms, notably allowing us to produce human insulin rather than having to extract it from the pancreas of different animals, and recently making the incredibly fast development of the COVID-19 vaccines possible. But how? Well, first, a restriction enzyme is used to split George's plasmid along a specific sequence of the DNA. The same restriction enzyme is used again to cut out the section of DNA coding for what we want George to replicate along the same sequence. Since the sequence both the plasmid and the section of DNA we're split along are the same, they can be stuck together using an enzyme called DNA ligase, reforming the circular shape of the plasmid with new DNA included. As much as we love George, he doesn't exactly have the brain power to know that we've added something to his plasmid, so he will produce the protein coded for by the new addition without complaint. Thanks, George.
You're welcome.